Hey everybody, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. If you are in my class, if you're watching this video for one or two reasons, you're either in my class, therefore learn the material the way that I write it on the board and what I want you to know, follow along in the note set. If you're not in my class and you stumbled across this in YouTube land, your instructor may have more detail or emphasize a little bit more of this and less of that than I do. Please make sure you learn the material the way that your instructors do. They want you to do it. But either way, hit like and subscribe so that when new videos come up, you guys can be notified. Now, again, these videos are intended for my students in my class, but I've had a number of people tell me they find them helpful. So anybody who wants to watch them can. Now, if you're in my class, we're going to be continuing our bone discussions. We've done a couple of lectures on bone tissue, what it looks like, what's the difference between compact bone and spongy bone. What we're going to be talking about today is on page 42 of my note set, bone development and growth. How bones develop is a process called ossification. And ossification simply means the formation of bone. So you should know that definition. You should be able to write that out. There's another definition in the note set called calcification. To calcify a tissue simply means you deposit calcium into the tissue. The difference between calcification and ossification is this. Calcification does not require any presence of proteins. So if I dump a bunch of calcium salts into a tissue and they form a hard crystal ball of calcium, we call that a calcium deposit. Calcium deposits typically do not have protein fibers growing through them like collagen and elastin and other things. But in order to form bone tissue, we need collagen fibers and elastic fibers growing in this network, and then we pack the calcium salts around that. And what those protein fibers do is they allow us to flex a bone without it completely snapping in half. It's kind of the same thing if you took a stick that's still part of a tree that has leaves on it, it's green wood. If you try to break it, the stick will snap, but it'll always have a piece holding it on. And it's kind of green in there, and what's holding it together and really hard to snap it is a lot of protein fibers growing in. And if you do break it, it rips a big chunk off as you rip along those protein lines. If you take a piece of dead wood, it snaps in two because all the protein has withered away and been digested after death of the tissue, which would also happen in bones. So, calcification does not require the presence of protein. It's just the deposition or the depositing of calcium into a tissue. If we deposit calcium onto protein fibers, then we're forming bone, which is called ossification. Ossification is the formation of bone. Calcification is just the depositing of calcium salts without the protein. Okay, you should know those differences. Now, when it comes to bone development, there's two types of bone development that we're going to talk about. There's going to be endochondral ossification and intramembranous ossification. We're going to write this stuff out in just a second. What I want to get clear to you guys, especially when we cover endochondral ossification, endochondral means inside of cartilage. So if I look at these two words, endo means inside, and chondral, as you know, chond always refers to cartilage. So in endochondral ossification, this is the formation of bone within a cartilage model. And we're going to go over this in great detail in a second, but essentially, during our development, when we're in the womb, we have, for example, like a miniature femur that forms, but the entire femur structure would be made entirely out of cartilage. And then within this cartilage model, we're going to remove some cartilage. We go in and remove some and put some bone in here. And then we remove more cartilage and replace it with bone. And over time, the bone will grow out from that center. So endochondral ossification simply is the formation of bone, ossification, within a cartilage model or inside a cartilage model of the bone. And this happens for all the long bones of the body. Now, one of the things I want to get clear is cartilage does not turn into bone. It doesn't. People often after my lectures come and ask questions. Hey, Mr. Long, you know when cartilage turns to bone? No. Cartilage does not turn into bone. We remove cartilage and put bone in its place. And I think of it in these terms. If I built a wooden fence and made a pen for animals, now, this wooden fence is not going to turn into brick. What I can do is I can remove a small section and put some brick in its place. Remove another section and replace it with brick. And over time, if I continue to remove pieces of wood and put brick in its place, eventually I would have a brick fence. The wood did not turn into brick. I removed the wood and replaced it with brick. Cartilage does not turn into bone. We remove cartilage and put bone in its place. Okay? And that's what happens in endochondral ossification. I can't find my eraser. Oh, sorry. There it is. So, now the other type of cartilage formation that we're going to talk about is called intramembranous. Intra always means within or inside also. It means inside like endo does. But in intramembranous ossification, and I'm just going to abbreviate the word ossification, this is the replacement of mesenchymal tissue with bone. I'm going to put in parentheses here, it occurs in the dermis of the skin. So the bones that form this way are called dermal bones, and dermal bones you also know as flat bones when we study the shapes of bones. So mesenchyme is embryonic tissue. So early in our development, some of our tissues are somewhat amorphous. They haven't really developed, but there's some embryonic connective tissue there. And within that connective tissue, some of the cells will morph into osteoprogenitors and osteoblasts and start to lay down some bone tissue, and it grows within the membrane. So endochondral ossification is when we form a cartilage model first and then replace the cartilage with bone. Intramembranous ossification is when we take some mesenchymal tissue and simply dump some bone in it. We replace some of that tissue with bone. And it's the dermal or flat bones of the body. So the bones of your scalp, there's not a lot of tissue separating the outside world from your bones. You can make an incision and it wouldn't have to go very deep before you hit bone. You don't have to cut a lot of muscle or a lot of other tissues. Same thing for your clavicle. You can touch it, and your ribs, your sternum. Now, for the long bones, there's muscle and bone and other tissues between the skin and those bones. So if you can feel it through your skin, it's probably intramembranous ossification. Okay, and it's a flat bone or dermal bone. Now we're going to go over the steps of endochondral ossification and intramembranous ossification. And as I was writing my note set or reviewing it, I realized my steps for intramembranous ossification, I don't like the way they're set up. So anyway, if you're following along in my note set, on the bottom of page 42, it asks you to draw out the steps of endochondral ossification. And these are done in your book in a series of steps. And, you know, a lot of times in A&P, we like to ask questions where you put the steps of things in the correct order so you understand the process. And you should do that for endochondral ossification. I'm going to make some drawings across here and give you, I'm going to shorten what's in the textbook into a sentence or two, because it's very difficult on a timed test to say, put the following 7 paragraphs or 6 paragraphs in correct order. So I like to cut it down to sort of a sentence or two for each step. Now, prior to all of this, what happens is we form a cartilage model of bone. And I'm just going to do sort of the old Bugs Bunny Roadrunner show kind of bone, nothing particular in the body. Okay, but this is a hyaline cartilage model of bone. It looks like a bone. When you take an x-ray of an embryo, some of the structures in there will make shadows on the x-ray that look like a humerus and a radius and an ulna and the carpals and metacarpals and phalanges and the femur and the tibia and the fibula and the patella. They look like the outline and the image of that, but they're not really made out of bone yet. They're made out 100% out of hyaline cartilage. Now, one of the things I did not emphasize is this. Some of it I did emphasize a little bit. Epithelial tissues are always avascular. There's no blood vessels in them. Most connective tissues are extremely vascular. Adipose tissue, bone tissue, most of the connective tissues that we've talked about have a lot of blood vessels growing through them. One exception is cartilage. There's a compound that is secreted in cartilage called Antiangiogenesis Factor. Now, I want you to just look at this word because, in my opinion, vocabulary is extremely important. Even if you don't know what something means, you can guess. Anti means not or against. Angio refers to vessels. So if someone has an angioplasty, they're having surgery on some kind of vessel. Or an angiogram is you inject dyes and you see where the blood vessels go. You're looking at a diagram or sort of an x-ray of all the blood vessels. And genesis means to make. So there's a thing called anti-angiogenesis factor. Okay? This is a compound that is found in cartilage tissue. And what it does is it prevents the formation of blood vessels in cartilage. And it can even cause the retraction of blood vessels from cartilage tissue. And because of this compound, cartilage is largely avascular, especially after we reach our adult growth stages. Okay? When we're young, cartilage has blood vessels in it, and if you tear the cartilage, it will heal because the blood supplies all the nutrients and compounds needed to repair the cartilage. But once we hit our teens and the anti-angiogenesis factor builds up, the blood vessels in cartilage will start to go away and retract. And that's why as we hit our teen years and older, when you tear cartilage, it's torn. If you tear the meniscus in the knee, it's torn for life. And there's not much you can do about it other than have the tear shaved out so it's not flopping around in there. And it's due to this compound. Now, oddly enough, just a side note, there's some research using this compound to cause the retraction of blood vessels in certain cancerous tumors. And so it may eventually lead to some treatments for cancer. They haven't perfected it, it's being worked on. But it's another avenue to attack cancer because cancer cells divide so rapidly and so much, they require a high volume of blood and nutrients and ions, and so they develop their own blood supply. And if we could inject this directly into the tumor, we could cause the blood vessels to retract. Would the tumor stop growing? And there's some evidence that maybe it does. Okay. Anyway, so anti-angiogenesis factor will eventually cause all the blood vessels that are growing in here to disappear. Without blood supply, the chondrocytes in the tissue cannot survive. They have to have nutrients. They have to have a food supply. And the oldest piece of cartilage is right here in the middle. And over time, we added more cartilage to make this cartilage model. The first place that the blood vessels disappear is where the oldest cartilage is. So right in the center of our hyaline cartilage model, the cells called chondrocytes, due to a lack of blood supply, will start to swell up, or what we say hypertrophy. Hypertrophy means to enlarge in size. So they start to swell up and get very big, increasing the size of the lacunae, and then the cells die, leaving these large empty spaces all within the tissue. So within our hyaline cartilage model, I'm going to end up with all these empty chambers near the middle. And that's kind of our first step of endochondral ossification. We can say that within a hyaline cartilage model, chondrocytes, hypertrophy, and die. And they leave these empty spaces in there. Now, there's a membrane that surrounds the cartilage called the perichondrium. I don't know if this light blue marker shows up very well, but that would be our perichondrium. When cells are dying, they release chemicals. Cells communicate by releasing all these chemicals, and some of these substances communicate to other cells that these cells are in trouble. And as they're dying and releasing those chemicals actually can stimulate the regrowth of blood vessels. And blood vessels will start to grow into and penetrate the periosteum and start to grow into the cartilage tissue. So one of the things that we will see happening next is that blood vessels will penetrate the perichondrium and grow into the middle and form a small collar of bone. And so our hyaline cartilage model will start to look like this. There's still some dead cartilage tissue in here, but what happens is there's a thin collar of bone that forms. And we would still have our perichondrium, but in part of it, because the cells are moving out forming bone here, it becomes periosteum. Where the light blue stuff would be our perichondrium surrounding the cartilage. Okay? Now, that happens because blood vessels are stimulated to grow in here by the dying cells. So I have some blood vessels penetrating the perichondrium, converting it into periosteum, and then some osteoblasts form a collar of bone. And that's really what's happening in our second step, is that blood vessels penetrate the perichondrium. This converts it to periosteum. And osteoblasts form a collar of bone. Now, in our third step, as the blood vessels continue to penetrate, osteoblasts will continue to move in, and really some osteoclasts also move in and eat away the cartilage, and then osteoblasts put down some spongy bone, and our model will start to look a little bit different. So we have a collar of some compact bone out here, but then we form a bunch of spongy bone in the middle. And all of this out here would be solid cartilage. And that happens because the blood vessels continue to penetrate here. So I'm having my different colors to represent the periosteum and the perichondrium. Our blood vessels have penetrated deep into the tissue here. But essentially, this little area of spongy bone in the center is called a primary, and we abbreviate a lot of times primary as a 1 with a little "ot" on it. It's called a primary ossification center. So I'm going to shorten this step up and simply say that in the center of the model, osteoblasts form a primary ossification center. And that ossification center is made out of spongy bone. So, within our cartilage model, some chondrocytes have hypertrophied and died. Blood vessels penetrate, they convert the perichondrium into periosteum, and osteoblasts form a thin collar of bone. As the cells move in deeper, they form a primary ossification center of spongy bone with all the trabeculae. Now, over time, the cartilage cells on the edge here, on the two edges of this ossification center, continue to die, and the bone growth will continue outwardly from that ossification center. So over time, it would start to look more like this. I would have a thin collar of compact bone, and this whole thing would be filled with spongy bone. And then out here, where the epiphyses would be, I still have some cartilage. And as that primary ossification center is extending outwardly, let me make sure I'm not getting too far to the edge of my screen. I'm getting close to it. But as the ossification center is growing out from this spot, osteoclasts will start to eat away the marrow cavity, or eat away some of the spongy bone and form the marrow cavity, leaving only a thin shell of spongy bone at the edge. So that is going to be our fourth step. As the ossification center grows, osteoclasts form a marrow cavity. So now you can start to see the beginnings of what looks like the diaphysis of a long bone. Now, I'm running out of board. I have a really large board here that goes another 15, 20 feet. But my camera, in order for you guys to be able to see what I'm writing, I have to have my camera in a certain spot. So I'm going to go back and erase the first step and the second step, and we're going to put the last two steps in here. Okay. Now, you need to be able to shorten this as much as possible, and then for memory, and then add in a little bit more detail. We have a hyaline cartilage model. The chondrocytes hypertrophy and die. Blood vessels penetrate the perichondrium. It turns into periosteum, and then osteoblasts move in and form a collar of bone. In the center of our hyaline cartilage model, some osteoblasts will form a primary ossification center of spongy bone. And then that primary ossification center will continue out. As cartilage dies, we replace the cartilage with some bone, and then osteoclasts start to form a marrow cavity, and it fills with blood and bone marrow. Okay? Now, over time, if you look at this very, very carefully, over time, what you will see is this. In the center of these two epiphyses, which are still a hyaline cartilage model, we're going to have blood vessels penetrate. I didn't draw the perichondrium or the periosteum on there, but they would be there. But essentially what happens is cells are still dying within the epiphyses, and just like we saw here, blood vessels will penetrate and form ossification centers in the two epiphyses, and we call these secondary ossification centers. So if I were to draw the picture, it would start to look something like this. I have a thin shell of compact bone, and then I fill this with spongy bone. I have where my primary ossification center was forming the diaphysis with its marrow cavity and spongy bone, and then the other epiphysis is a thin shell of compact with a lot of spongy bone. And of course now the membrane all the way down is periosteum. But essentially what has happened is this. Blood vessels have penetrated all along here, but then they penetrate into the epiphyses. And that's essentially what's going to be our next step. we can say chondrocytes die within the cartilaginous, and you don't have to put all these words down, but I just want to get the idea across to you, within the cartilaginous epiphyses. Okay? I'm going to put this in parentheses sort of as a reminder of what's happening. Within these epiphyses that are cartilage, the chondrocytes are dying. But essentially, we can simply say this. This is the main thing. Blood vessels penetrate the epiphyses and form what we call secondary. This 2' simply means the word secondary. And we have secondary ossification centers that are formed within the epiphyses. So, blood vessels penetrate the epiphyses and form secondary ossification centers. And that's because chondrocytes were dying in there, and the cartilaginous epiphyses get converted into, we remove cartilage and put bone in its place. Well, essentially what happens while that's occurring is that we leave a little bit of hyaline cartilage in the growth plates, and we leave a thin edge of hyaline cartilage on the articular surface. And this looks like the drawing of the long bone that we did in one of the first lectures. Spongy bone in the epiphyses with a thin shell of compact bone. The growth plates or epiphyseal plates or metaphysis is hyaline cartilage. We have some articular cartilage on the edge. And then we have our diaphysis with the marrow cavity. And we could put a last step on here. Essentially in step 6, all that's going to happen is we're going to continue to remodel the bone. So we can say bone remodeling continues, and we will remove some spongy bone and replace it with some compact bone and remodel the bone for the rest of your life. Okay? Now, bone growth and development begins about 6 weeks after fertilization when the sperm meets the egg. Within about 6 weeks, we're starting to form cartilaginous little cartilaginous models of bone as an embryo. And that's going to continue until we're somewhere in our late teens to middle 20s. Our skeleton can continue to grow and get larger from birth all the way up until we're somewhere between like 17, 18, and about 25 to 27. That's when we're done growing and forming our skeleton. For females, it's usually a little sooner or earlier in life. Males continue to grow and develop their skeleton a little longer due to our sex hormones and some other things that we're going to talk about. So, I hope all of y'all can learn these steps of endochondral ossification, can draw it out and teach it to someone else. Imagine if you had to give a speech to a 6th grade class on endochondral ossification. You need to explain to someone what is ossification, and then you need to draw out the steps and be able to explain it, or at least cut it down to these sentences, okay? So, I'm going to cut it for this lecture, and we'll do another lecture on intramembranous ossification and bone growth. So, I hope you learned something. I hope you had as much fun as I did. I want you to continue to do this until you can't stand it. Do it till you understand it, and then do it 5 more times so that you can make an A on your tests, okay? You should know the steps of bone growth and development. And these steps are going to be important when we cover a lot of hormones in part 2A and P, and they're going to be important for bone repair or fracture healing, which we're going to discuss in another lecture. So, thanks for watching, and again, I hope you had as much fun as I did. I'll see you in the next video.